Good afternoon, all. Previous lecture, I discussed with chemical structure of the nitrogen base, pentose sugar, and phosphoric acid. Now, today, I will tell you in brief that how the nucleosides are formed and nucleotides are formed. You know that nucleoside means pentose sugar and nitrogen base. So here pentose sugar and nitrogen base are fused together and form the nucleoside. So here two types of nucleoside. Deoxyribonucleoside and ribonucleoside. Yes, deoxyribonucleoside in that deoxyribose pentose sugar is associated with the nitrogen base, purine as well as pyrimidine. So, if you are talking about the purine, please revise little bit. You revise that. If I am talking about the purine nitrogen base, so ninth number nitrogen is attached with the first number of carbon of pentose sugar. Again, if you are talking about the purine nitrogen base, ninth number nitrogen of the nitrogen base and first number carbon of the pentose sugar by dehydration process. Yes, ninth number carbon over that hydrogen is attached and first number carbon OH is attached. H and OH join together and release the water and bond is created. Okay, so deoxyribose sugar is designated by sign in this way. See, this is the deoxyribose sugar sign. You know that over that oxygen is there and here on the first carbon nitrogen base is attached. So here adenine is attached. Here deoxyribose sugar with this guanine is attached. Cytosine is attached. And thymine is attached. But remember, with deoxyribose sugar, uracil is never attached. Got it? So if you are talking about the ribonucleoside, so ribonucleoside, ribose sugar is there. It is denoted by this R. Yes, now all the structures are same. Adenine, guanine, cytosine, uracil. But remember, here with ribose sugar, thymine is never attached. Okay, so in this structure, you see in the structure, pentose sugar, over that nitrogen base is attached on the first carbon. Fine. So if you are talking about the pyrimidine nitrogen base, so pyrimidine nitrogen base, first number nitrogen of the nitrogen base and first number carbon of the pentose sugar is attached. So hope you understand this. Now nucleotide. Okay, let's see the nucleotide. Nucleoside plus phosphate group, or you say that pentose sugar, then nitrogen base and phosphate group. Okay, so again here same way, nitrogen base, sorry, nucleotide is of also two types. Yes. So same way deoxyribonucleoside, here deoxyribonucleotide is there. Same way ribonucleoside, here ribonucleotide. What is the difference only? Here, remember dear all students that phosphate group is attached on the fifth number carbon of the pentose sugar. If you revise the structure, fifth number carbon on the pentose sugar is over the CH2OH side. Okay, so in symbolic manner, deoxyribose sugar with adenine, this is known as nucleotide, sorry, side. Now what is tide? So here over the fifth carbon phosphate group is attached. This is known as deoxyribonucleotide. Now same way here we have to draw all the structures, and over the fifth carbon I have to put the phosphate group. So again deoxyribose sugar, adenine, then here guanine. Here phosphate group is there. Cytosine, here phosphate group is there. Thymine, here phosphate group is there. Here it is not possible at all. deoxyribose sugar with uracil and phosphoric acid. Fine. Now same way here, ribonucleotide, ribose sugar with adenine, and here fifth carbon phosphate group, ribose sugar, guanine, phosphate group, ribose sugar, cytosine, phosphate group, ribose sugar, uracil with phosphate group, and last one, ribose sugar, uracil with, sorry, mistake here, this is the thymine and here it is not possible. Dear all students, now please make concentrate here. I can now draw the only one. If you have to do the practice, then you can carry on. I have no problem at all. But only I will draw the one structure of nucleoside and one structure of the nucleotide. Okay? Fine. Just for example, deoxyribose sugar with adenine. चलो फाइन आ फर्स्ट ज ड्रॉ करू छू सो यू नो दैट deoxyribose sugar okay, see carefully, you see this is the deoxyribose sugar, here H is there. Now as you know that I drawn the diagram, the structure of adenine. So you know that adenine formula 1 amino, right? So here 1 amino, 2 alternate bonds, here 1 hydrogen filled, here 1 hydrogen filled, and here 1 hydrogen filled. Okay, you know that what is nucleoside? See here what happened, that this nitrogen from H and this carbon from OH, it is released by removal of the water. Okay, and here this is known as glycosidic bond. Okay, So here from ninth number nitrogen, hydrogen, and from first number carbon OH, both are joined together, water is released. So this molecule is known as, so just I am making arrow here in this way, that this is the structure of nucleoside. Dear all students, now you understand that this is the pentose sugar, deoxyribose sugar denoted by this, and adenine sugar is denoted by this. So if you draw the now here nitrogen base as a guanine, you can make also practice like that. This is also very fine. And here instead of this purine nitrogen base, you draw the pyrimidine nitrogen base also. Okay, so this is the first one example of the nucleoside. Now, what is nucleotide? See, here I had already taken the adenine example from DNA. Fine. Deoxyribonucleotide purine nitrogen base I have already taken. Suppose example, if you want to draw the this one, fine, deoxyribose, cytosine and phosphoric acid. So first I will draw the deoxyribose sugar. This is the deoxyribose sugar. Now cytosine. Cytosine 1 amino 3 keto. Yes, you know that now here bond is formed, nucleoside, and here phosphoric acid I am right here. Fine. Okay. Now what you understand here, from nitrogen H is released and from carbon OH is released. Here one bond is formed. Fine. And here from CH2OH, OH is released, and from OH H is released. And now here I can change little bit. CH2 I can write here, but carbon form the bond with oxygen. Okay. So here ester bond is there. Here ester bond and here glycosidic bond. So here two water molecules are released. Here one water molecule is released. Okay, so now Listen the next sentence carefully. Okay. Now see, this is the very important one. When two nucleotides means same, if you use the deoxyribonucleotide, then you have to use the same nucleotides on that strand. Okay, someone says that sir, I can use the deoxyribonucleotide and next can I use the ribonucleotide? No way at all. If you use the deoxyribonucleotide, then you have to use the same one. Same way, if you want to use the ribonucleotide, then you have to use the same. Fine, but this is the most important one. The two nucleotides are joined together in such a way that what? Third carbon of pentose sugar. Wait. I can now draw here. Two nucleotides I am drawing here. Wait. This is the pentose sugar. Deoxyribose. Suppose with that adenine is there. Suppose here with deoxyribose sugar, adenine is there, and with this deoxyribose pentose sugar, cytosine is there. These two are known as nucleoside. Now, if I will put phosphate group here and here, known as nucleotide. Now here you assume, see, you think about, on the first nitrogen base 1, 2 and 3, third carbon, and when you see the pentose sugar 1, 2, 3, 4 and 5, here the P is the 5 one. Okay, so third carbon of pentose sugar of first nucleotide and fifth carbon of the pentose sugar of second, means next nucleotide. So what is this? Here this one. As you know that these are glycosidic bond and these are ester bond. Now this bond is known as phosphodiester bond. In this way, two nucleotides are formed. In same way, the two nucleotides, so third nucleotide, fourth, and many nucleotides are joined together one by one and form the long strand, and that is known as polynucleotide. Okay, so same way polynucleotide, it is of DNA as well as RNA also. So this is about the polymer. Okay, let's see the primary and secondary metabolites. The most exciting aspect of chemistry deals with isolating thousands of compounds, small and big, from living organisms, determining their structure and if possible synthesizing them. એ ક્યાંથી ફોર્મ થાય છે, that is also important. If one were to make the list of biomolecules, such a list would have thousands of organic compounds including amino acids, sugars, etc. For reasons that are given in section number 9.10, we can recall these biomolecules as a metabolites. Yes, carbohydrate, protein, lipid, all these molecules are also known as metabolites, and for them the process is there known as metabolic process. Metabolic process is also known as physiological activity. And you know that we had already learned in the first chapter that metabolic activity is carried out in the two phases, anabolic and catabolic. In animal tissue, one notices the presence of all such categorized compounds shown in the figure number 9.1. These are known as primary metabolites. So move on the page, previous page, primary metabolites 9.1, carbon, hydrogen, oxygen, nitrogen, sulfur, sodium, calcium, magnesium, like that. However, when one analyzes plants, fungal and microbial cells, one would see thousands of compounds that other than these primary metabolites. So if we observe the other animals cell, plant cell, fungal cell, bacterial cell, then we obtain other than the primary metabolites, other than the nitrogen, hydrogen, carbon, oxygen, sodium, sulfur, magnesium, silicon, like that. If other compounds, if we observe, these are known as secondary metabolites. Alkaloids. These are the examples of the secondary metabolites. Alkaloids. See, which are the alkaloids? See in the table. Morphine and codeine are known as addictive drugs or narcotic drugs. Okay, here morphine. Yes, write that morphine is painkiller. Used in World War. The injured soldiers can use the morphine. So morphine acts as a painkiller. Fine. Codeine. Codeine used in cough syrup. It decreases the stress for coughing created in alimentary canal. So morphine and codeine are obtained from opium. So these are opioids. बराबर. Morphine and codeine are opioids. એને અફીણ કહેવાય. This is extracted from the plant. Flavonoids, rubber, essential oils, antibiotics, colored pigments, scent, gums, spices. These are called as secondary metabolites. See in the table 9.3. Pigment. You know, carotenoid. Carotenoids are yellowish to orange colored pigments. Carotenoids include carotene and xanthophyll. Again, carotenoids are known as yellowish to orange colored pigments. Carotenoid consists carotene as well as xanthophyll. So carotene is orange colored, xanthophyll is yellow colored. Yes, anthocyanin, it's bluish colored. Then alkaloids, I already told to you. Terpenoids, monoterpenes and diterpenes. Terpenes obtained from turpentine. Essential oils, lemongrass oil. Toxins, poisonous substance, heparin and ricin. Lecithin, lectins. Concanavalin A. Drugs, venblastin, curcumin. Polymeric substances, rubber, gum, cellulose, pectin, lignin, like that. While primary metabolites have identifiable functions and play no role in the normal physiological processes, we do not at the moment understand the role or functions of this secondary metabolite in the host organism. When primary metabolites can function it properly in the body, then we can't see the function of the secondary metabolite. It's a very complex procedure. That's why I use the word metabolic traffic in the body. However, many of them are useful to human being: rubber, drugs, spices, scent and pigments. Yes, chlorophyll is green colored pigment and it is converted into yellow, orange, blue, red, like that. Okay, and you had already studied in 8th chapter. Okay, chromoplast, chloroplast, apart from that, chromoplast, different colored pigments. Food storing molecules, colored pigments, you had already studied with this. Some secondary metabolites have ecological importance. In the later chapters and years, you will learn more about this. Now, biomacromolecules. There is one feature common to all these compounds found in the acid soluble pool. Acid soluble pool means that some substances are dissolved in acid. The substance is commonly known as acid soluble pool. They have molecular weight ranging from 18 to around 800 Dalton approximately. 1 Dalton is equal to, please remember, one Dalton is equal to 1.67 into 10 raised to minus 27 kilogram. Okay, so the molecules which have the molecular weight range between 18 to 800 Dalton. These all molecules are including acid soluble pool. Acid insoluble fraction has only 4 types of organic compound. Now these are acid insoluble, that is protein, nucleic acid, polysaccharide and lipids. This classes of compounds with exception of the lipid, because remember all the molecules are included in acid insoluble pool except the lipid, because please underline, lipid has molecular weight more than 10,000 Dalton, because it has molecular weight in the range of 10,000 Dalton and above. So all students, please remember here two types of molecule, acid soluble and acid insoluble. Acid insoluble compounds in that we include protein, nucleic acid, polysaccharide and lipid. But here with the exception of the lipid. Why? Because lipid consists greater than 10,000 Dalton molecular weight. For this very reason, biomolecules, that is chemical compounds found in living organisms, are of two types: micromolecules and macromolecules. Those which have molecular weight less than 1000 Dalton. Okay, fine. So the molecules which have molecular weight less than 1000 Dalton are included in micromolecules and are acid soluble. Are acid soluble. While those which are found in acid insoluble fraction, okay, are known as biomacromolecules or macromolecules. So here the molecular weight Okay, so here please remember, except the lipid, because you know that lipid consists more than 10,000 Dalton molecular weight. Okay, so here I can stop. Remaining things we will see in next lecture. Thank you very much to all. Hope you understand this. See you in next lecture.